Hello. In this video, we are going to talk about how HIV is transmitted and how it's so sinister, like how it affects your immune system and results in AIDS. Remember, HIV is the virus, but AIDS is the resulting disease because HIV attacks a very specific cell in your body called the T cell. And if you remember when we were talking about antibody production, those T cells are vital to give the information to the antibody producing B cells what pathogen is in your body. Just a couple, let's clear up a couple misconceptions first. Um, a couple things. HIV is not transmitted by mosquitoes. If a mosquito bites somebody with HIV and then bites you, you're not going to get HIV. It's very hard to get it from kissing unless you actually bite the person and you're also bleeding. Definitely not swimming pools. You can't get it from sitting on toilet seats. Holding hands is perfectly fine. And unlike other viral infections, like Corona and SARS and all the other ones out there, it's not transmitted through coughing. One more misconception I want to bring up is HIV is a zoonotic disease. Zoonotic means that it was in one species and then it jumped to another. And it did jump from a species of monkey to humans. That is totally true. However, a lot of people have this idea that there was some relations going on between humans and monkeys, and that is not what happened. Because in some parts of the world, in Africa, it's traditional to have monkeys as part of your diet, and in the cleaning process and the killing process, there was blood-to-blood contact. HIV is, however, passed on through three main ways. So definitely pregnancy and breastfeeding, believe it or not. So that's why a lot of times if a mother has HIV, she'll pass it on to her child. And they can do things to try to suppress the HIV during childbirth to make sure that baby does not get HIV. Number two, definitely sex is one way that HIV can be transmitted. And another very common way is through sharing hypodermic needles, especially during like drug use, because quite often people reuse needles when they're doing narcotics and illegal drugs. So those are some misconceptions I wanted to clear up. Let's go to the next slide. So I want to take a second and just talk about the specifics in how HIV is transmitted. So in this diagram, this is the virus. It actually looks kind of like a coronavirus. A coronavirus has these proteins on the outside of it, and these proteins allow it to bind. So actually, step one is called binding. I'm going to make that a little smaller. So binding occurs when receptors on the HIV virus match receptors on your cell, and specifically, it's your T cell. So this big orange yellowish cell is supposed to be a T cell. So there's receptors. You don't need to know the specific receptors unless you're going to go into epidemiology, but they're called CD4 receptors, and the virus binds. When the virus binds, it releases its genetic material into the host cell in the T cell. So some viruses release RNA, some release DNA. So the HIV virus is an RNA virus, and it's going to carry out a process called reverse transcription or reverse transcriptase. So the RNA that's in the virus will be converted into DNA. So if you remember, transcription was taking DNA and converting it into RNA, and reverse transcription is going the other way. We're taking that DNA, or we're taking that RNA that the virus injected into the T cell, and we're converting it to DNA. The third step is called integrase or integration, and that's when we take the virus's DNA, where you can see it looks like a figure 8. I'm not quite sure why they did it this way. I guess it's supposed to be helix. But the virus's DNA gets integrate into the human DNA. And there's been some studies to analyze the human genome and estimate what percent of the human genome is viral, because here you have virus DNA in there, in this little yellow area. What percent is viral? Now, there's a couple things that have to happen. So through the course of evolution, if, and these are like a lot of ifs, but it happens, right? Because humans have been around for a while. If this DNA was inserted into a gamete, so we're talking either an egg or a sperm, and then that gamete fertilizes another gamete, so fertilization takes place, then this DNA will survive. Usually what happens if it's not a sex cell is this virus will eventually destroy this cell and the DNA will just disappear. But if it's a sex cell and we can carry that genetic information before it happens, you can incorporate the virus DNA into human DNA. And the estimates are about 14% of human DNA. Let's try that again. 14% of the human genome consists of viral DNA. So here we have DNA in our own chromosome. Then transcription occurs, just like normal transcription. So transcription is the production of RNA, messenger RNA. I'm going to be lazy and just write this. So we've made a piece of messenger RNA, but it's not human, right? The messenger RNA has the information from the viral genome. So your cells are like la-ti-da-ti-da, just copying the DNA. However, we've made accidentally the code for producing viral proteins. So this, if you remember how ribosomes work, this messenger RNA will go to a ribosome, and then we're going to begin producing amino acids right off the top. And these amino acids are going to eventually form the proteins. So we have transcription and translation occurring. So translations occurred. We've got viral parts of proteins made. Then they're assembled. They're all put together. We get some, it steals some of the phospholipids that you have inside of your cell. The proteins are assembled. It all comes, what's so crazy about viruses, they have enzymes that they release in here as well that help with this assembly process. So that's the next phase called assembly. So now we have a virus completely assembled and just waiting to escape. Now, you may have heard with HIV, it can stay inside these T cells. Like, people may not show the symptoms for quite a long time. Like, I think it's like 6 months, 3 to 6 months. So until the viruses escape from this cell, you can't infect anybody, and people don't show symptoms. So we have the assembly process. The next phase, number 6 here, is called budding, where the virus begins to bulge off of the T cell. The T cell loses all of its contents and dies, and now you have a fresh new virus that can go on and attack another T cell. And this is how your immune system is destroyed. So what I'm going to do is just go to another slide, just to show you a little bit of a graph to explain like what happens here. So there are two lines here. So the red line is talking about um, the amount of HIV like made in your blood, and the blue line is the number of T cells in your body. So here's where the infection occurred, up here. So it looks like week maybe 3. So in the beginning here, you're getting lots and lots of copies, and then as it peaks, your T cells begin to decrease. So I was wrong about the 6 months. It looks more like 3 weeks. Then it goes like into a sleep period, and it can stay dormant inside of your body for quite a long time. However, they're living and destroying all these T cells, so um, the number of T cells in your body slowly decrease and decrease. And when you get below this critical level, it looks like the number is 200 cells per probably cubic micro. That can't be right. Cubic micrometer. Yeah, maybe millimeter. Then that's just too low. And then you get all of these illnesses because you don't have the T cells to protect you. And eventually it leads to death. The medicine they have now is designed to keep the HIV inside of your T cells so they don't leave. So people can live a very normal, healthy life. One of the drugs is called ART. It stands for anti-transcription, anti-reverse transcription. Right? So you're not allowing that reverse transcription to happen that we talked in that previous slide to it to happen. PEPFAR is another one, but all of these work out really well to slow down the effects. So pretty sinister, pretty evil how this virus works by just targeting your T cells, eventually leading to a decrease in them, and then you can't find infection. You can't fight infection, and eventually it leads to death.